Hello, I am Amelia Shebnell. For the new students who are on here, I am a geological oceanography professor, and I am running the seminar series for this year. And for this semester, at least, we are going to have a remote seminar series, and we're trying to bring a little bit of normalcy to our week. We usually have these seminars each week on Friday at this time. And so I encourage you all to attend throughout the semester. We have a really great slate of speakers. I'm really excited. I think we got people who we wouldn't normally get because everybody's trapped at home. We are also having a meeting for the students and the speakers each week if the speaker is available. We are putting together that schedule right now. Most speakers are able to meet at 2 p.m. our time for the students. So that's after professional development. We have a couple of speakers who have their own weekly seminar in that time block, and so they actually have to go at noon. But right now, I think that's only like 1 or 2 speakers. So I look forward to seeing you all in seminars in the future, and I'm going to turn it over to Brad, who is going to introduce our seminar speaker this week. So thank you all for coming, and I hope you enjoy the seminar series.
All right. Thank you, Amelia. It is my pleasure to introduce our speaker today, Andreas Fahlman. I was virtually introduced to Andreas Fahlman about 2 years ago by Ken Story, who some of you may remember gave a talk here a few years back, an entertaining character who spent much of his seminar making fun of me. But he emailed me and told me that Andreas had moved to Florida recently and that we were both pirates, in his word, and we would probably get along well. And I think by Ken Story's definition, all of us are more pirates than he'll ever be anyway. But we did manage to connect, and that has resulted in a courtesy appointment here at USF, which I'm very happy about, and we're working on proposals and such things. Andreas got his bachelor's degree in '96 from Hawaii Pacific University, and went on to a PhD with Ken Story co-supervising at Carleton University in Ottawa, Canada, and he was working on the physiology of hydrogen diving and its implication for hydrogen biochemical decompression. Since then, he bounced around in a number of different research appointments in South Africa, in the States, in started to write it down, but British Columbia, Scotland. He's been all over the place before taking a position at Woods Hole in 2009. He was a research specialist there, and then took an assistant professorship at the Texas A&M University in Corpus Christi from 2011 to 2016. He is currently a senior researcher in Valencia, Spain, working on ecophysiology and conservation research. So without further ado, I should also point out that he has been extremely productive. According to the CV I had from a year ago when he was taking the courtesy appointment here, just over 100 publications. So that's quite a landmark, and I'm sure you'll all be entertained. So without further ado, Andreas, take it away.
Thank you very much, Brad. Thank you very much for this invitation. I'm sorry I missed being with you guys and actually talking to you in person, but we'll do what we can with this. So this is a journey, and I'm not sure that this, I think this is the first one. So I'm going to take you on a journey. This is about 12 years worth of data, I think, that has led to new hypotheses how marine species work when they're diving. So basically, I'm a comparative physiologist, and what we're trying to do is we're trying to understand how animals are functioning in their environment. And what you see here is a sea lion, and what most of them do is they prey on some type of species. And a lot of these species are temperature sensitive. So as you all know, with global warming and with climate change, the surface layers of the water is going to heat up. So what are the fish going to do? They basically are ectotherms, so they need to either move further down the water column, or they need to move to higher latitudes. And what we're trying to understand is what are the consequences for these animals to these changes in their environment, and how is that going to affect their foraging efficiency? How is it going to affect buoyancy, for example, because pressure will act on them, metabolic costs, gas exchange, and we're especially interested in looking at how the lungs are functioning and the effect of the lungs and gas exchange on decompression sickness. So for those of you who are diving, you've heard this before, and I'll explain this a little bit more. So this is much of what I've been doing and followed the footsteps of this gentleman. This is Per Scholander's seminal work from the 1940s. So this was written during the Second World War, and it was published in an obscure Norwegian journal, where he outlined some of the diving physiology of these animals, so turtles, marine mammals, seals, and whales, and how they're functioning. So one of the things he suggested was that these animals have specialized adaptations that avoid them to allow them to avoid decompression sickness. So for those of you who have taken physics, if you haven't taken physics, remember that the pressure-volume relationship when you're going down into the ocean, or if you're flying, but the relationship is slightly different. If you're going down into the ocean, for every 10 meters, the pressure increases by 1 atmosphere absolute. This then on a gas volume is reducing the volume and increasing the pressure. So if you're at 10 liters at the surface, you're at 5 liters at 10 meters, you're at 3 liters, you're at 2.5 liters at 20 meters, and so on. And this has an effect on these animals because the pressure on the chest, on these gas-filled spaces, is then compressing them. So what Scholander suggested was that these animals, they have these adaptations. So they have a non-collapsible dead space and a collapsible alveoli. So this balloon, we call it the balloon pipe model. So when they're going down into the ocean, the chest is compressing, and then eventually the alveoli, these small gas-filled balloons at the periphery of the lung where gas exchange occurs, will collapse, and there will be no gas exchange. So what will happen then is these animals will not take up any oxygen, they will not give off any carbon dioxide, and they will not take up any nitrogen, which is this gas that then leads to gas emboli, to bubbles, and decompression sickness. So this has since the 1940s been the working model how these animals are working and how they're functioning. And it's basically been a one for all animals are the same, all marine mammals are the same. So in the beginning of 2000, at the turn of the century and the millennium, there were some reports, and there had been some reports that there were strandings, there were stranded whales that did seem to have symptoms of decompression sickness. These were similar to the symptoms seen in divers and in animals that have been used in diving research. So then we were asking, what is actually going on, and how much of this hypothesis is actually true, because there hadn't been a lot of research on this adaptation that Scholander presented in 1940. So we were asking, his hypothesis assumes a rigid non-collapsible trachea, it assumes a compliant rib cage, and is it true that all mammals have these adaptations, and how much species difference are there? So do these species differences then cause some species to be more at risk or others not at risk? And then if they have certain behaviors, if they dive to certain depths, deeper, shallower, longer, shorter, is that increasing risk? And an important question is, at what depth do the alveoli collapse, and how does that affect gas dynamics? So how and where do they stop taking up this nitrogen? So we started looking into the literature, and this was published in 1970. This is an absolutely amazing feat of experiments. I don't know if you can see my cursor here, but what you see here is you see X-rays through a hyperbaric chamber. This chamber was rolled into the UC Davis Hospital and the University Hospital, and what you see here is this is a Weddell seal, and you see the trachea here, the outline of the trachea here and here. This round thing here is an ECG disc that sits on the outside of the seal. Here's the same animal breath hold diving to 300 meters. And you can see how the trachea, this upper airway, is compressed. So we took this to be that, look, this hypothesis is not really true. There's some caveats to it. So maybe the upper airways are stiffer, but they're not non-collapsible. So we started looking into this, and we tried to then understand how compliant or how collapsible are these tracheas. So on the left here, you see the trachea of a gray seal. On the right, you see a common dolphin. And I was telling the students before that this dolphin is 200 kilograms, so about 440 pounds. This gray seal is about 40, 60, maybe 80 pounds maximum. And you see the differences, this yellow ruler, you see the differences in length of the trachea. So they're huge species differences. And then the compliance of this, or what I call the squishiness when I explain this to my mom, is basically how plush are these? So I try to, if you squeeze on them, are they more stiff like a tube, or are they more like your pillow at home? Can you basically compress them? So we were trying to understand this and understand how compliant or how flexible are these. So we looked at different animals. So on the x-axis here, you have the initials for the genus and species. So on the left-hand side, you have a number of species of seals. You have odontocetes, which are toothed whales in the middle. You have sea lion, which is just a California sea lion, and then you have a couple of land mammals. So if Scholander's hypothesis was true, you would expect all the marine mammals to have much stiffer or less compliant upper airways. So the compliance would be much lower for the marine mammals than the land mammals. And you see there's a lot of variation here. One interesting thing was the effect of age. These are in gray seals. It increased, the compliance increased with age, which you would expect to be the opposite, because you would expect, if this has something to do with diving, older animals dive deeper, and then you would expect that the upper airways would be stiffer. So this lung collapse would have a better effect, and the lungs or the alveoli would collapse at lower depths. There were at least one species that had read Scholander's thesis, and these are the beaked whales, or the deep divers. So they had extremely stiff upper airways. So this was what it was expected in a very deep diver. So here the lungs would collapse at shallow depths because the upper airways were really, really stiff. Then what we did was we looked at the lungs themselves. These are, again, flow volume loops or compliance loops. So you see the solid line is for inflation and the dotted lines are for deflation. So what you do here is you inflate something, and then you see what the pressure difference is between the inside and the outside, and then we compare this with the total volume that was inflated, and we've inflated this to a standard volume that depends on the size of the lungs. You can see here some of the differences on the left-hand side in the seals. The seals have very compartmentalized lungs, and I will come back to that later on because this is important in this new theory. Versus the cetaceans or the whales. The whales have two lobes, one on each side, left and right, but they're completely solid. If you look at the compliance curve, so when the animals are diving, you would expect that these would be deflation curves, and you see how differently they are in the odontocetes, so the toothed whales, compared to the phocids, so the seals and the sea lions. So there seems to be some architecture that is very different in these species. So working at Woods Hole, I had the opportunity to work with a fellow called Michael Moore, not the documentary filmmaker, but a veterinary pathologist who is and still is like MacGyver, absolutely phenomenal. And so what we did was he had a grant that I was able to work with him on, and we built a hyperbaric chamber. This is basically a fiberglass tube, a long fiberglass tube here with end caps. So we water fill this, put cadavers inside, and then we could pressurize them to different pressures to see how the pressure were affecting the lungs. So these were the reason why this sits on a frame here is because this weighed about 3,000 pounds, because we needed to water fill this, and the table here is a human CT scanner, and it could only tolerate about 400 to 500 pounds. So we need to build a separate frame for it. What we then could do is what you see here is this is a harbor porpoise. On the left hand side you see the harbor porpoise and you see the lungs, and with the black outline you see the same animal at 50 meters and then at 100 meters, and you can see this gravity dependence of the air moving to the higher parts. This is a bit weird, and I'll come back to that. I'll show you some of that on the next slide where we do some comparisons between species. But I will come back to this, and remember this slide. Look at how the gas is moving. So you have a collapsed region down here, you have an open region up here. So it seems to be two regions in these species. Also look how the chest is compressing. So the chest is very compliant, very flexible. So it squishes inwards. Here's this species comparison. So on the left-hand side you have a dolphin, on the right-hand side you have a gray seal, and on the top here you have a pig. So this is a land mammal. Look again here. So these animals, this was some difficulties we initially had, because once you start, this chamber was water filled, and once you start compressing it, they changed the buoyancy. So these animals were floating around until we figured out that we could actually suction cup them to the hull of the chamber. So they're floating around and turning a little bit, which then turned into quite some interesting findings. So look here where you see this is a dolphin at about 40 meters, 50 meters. Here's the lungs are open here. This is the most, the highest point on this animal, while down here it's compressed and collapsed. Same thing with the gray seal. The lower regions are collapsed, the upper region is open. The pig is also tilted to the side, but you see that there's hardly any compression and collapse, and these are about the same pressures for all of them. There's also not a lot of compression on the chest, which is, land mammals are the very different chest. So compression and collapse does not occur in the land mammals the same way as it does in the marine mammals. So structural and functional variation between species, we showed that quite clearly. Alveolar collapse, we concluded from these studies, is probably much steeper than previously thought. The previous estimate had been about 30 meters for the seal and 70 meters for dolphin, but we estimated about 150 to 200 meters. Luckily, some smart people then actually did empirical measurements and have shown that in the sea lions, it's probably around 150 to 200 meters. And the variation in the collapse depth depends on the structural properties, and then it may affect diving capacity in different species. And then that may have an effect on how climate change will affect their ability to continue to diving deeper and for longer periods of time. So you may have been thinking, oh, these are all dead animals, that can't mean a lot. So we thought the same thing. Someone told me that you've been working on dead animals now, maybe you should work on live animals. So we started working with, this is from San Francisco, this is the Marine Mammal Center outside San Francisco in Sausalito, where they rehab animals. So sometimes they didn't need to do interventions, so we could, when they intubate the animals to anesthetize them, we could then do some measurements on these pressure-volume relationships. This was Vancouver Aquarium, where they have stellar sea lions, and sometimes they do procedures on these animals. These animals were working a lot in the open ocean and then came back and they stayed inside the aquarium at night. So when they were doing medical procedures, they anesthetized them and we could then do some measurements. what we then found, this again is a pressure volume loops. So the steeper this is, the squishier it is. So here you see the chest, they have very compliant chest. If you ever touch a seal or a sea lion, they're like a plush pillow. The lungs are less compliant. So it's basically the lungs that are setting how much the chest and the alveoli will collapse. So the chest itself does not cause any resistance to compression. So lung collapse in these animals and these species completely depend on the compliance of the lungs themselves, exactly what Scholander was suggesting. So in the pinnipeds, this seems to be the case. Something interesting came out, and this also been shown in humans, something fairly similar, that with lung function training, and basically what these animals are doing, they're diving every day and they're compressing and collapsing the lungs, and then the lungs are opening up, and then they're compressing the lungs and they're opening up. So you see here the stellar sea lions that lived and were born and raised in a zoo, in an aquarium, had much lower compliance. So, and the animals in the wild, so these were wild animals that were in a rehab center, had much higher compliance. So one of the hypotheses we came up with, the reason for this was maybe that these animals that are continuously diving have much more compliant lungs. So the lungs are squishier because this is an adaptation to deal with the pressure that they then are exposed to day in and day out. Well, when you're living in an aquarium, you don't dive and do not have to dive as much. This changes in compliance may have some clinical significance and clinical relevance, because a lot of people that have respiratory diseases may have stiff lungs. So if you do certain exercises, you potentially could then increase your lung compliance. So without taking drugs, you could potentially change the capacity for people with certain lung diseases. Then we started to think about maybe we should do some lung function testing on wild animals, and we went to New Zealand. And I'm sure you've all had done failed experiments, and this was one of mine, one of our greatest failures. We tried to do lung function testing on sperm whales, and you see that the party hat we tried to put over the blowhole is just way too small, and it was really scary to do this. So we thought maybe for our sanity and to actually get some data, we start working with aquariums and with facilities that house animals in human care. So we developed this pneumotachometer. Basically, it's a spirometer or a flow meter. So you see it here, place of the blowhole on a dolphin, so we could measure the respiratory flow, we could measure the respiratory gases. You see here during a trial where we do lung mechanics testing. You get to go to Hawaii to do this, so that's at least this initiative. Here the animal is taking an esophageal tube, so we can look at the esophageal tube, and that allows us to measure the compliance of the lungs and the chest in real time in the animals while they're sitting there. So this is what the data looks like. On the left-hand side, you have flow rates in liters per second. You have tidal volume, which is the integrated flow rate on the right-hand side, and then end tidal oxygen on the right-hand side. So I'm just going to show you some data and what it looks like. This is what it looks like when these animals are breathing. They oftentimes, if you listen to a dolphin or to a whale expire, it's like an explosion. They have really, really amazing respiratory capacity. So they can generate flow rates that are incredibly fast. A human is much lower. Your maximum capacity is about 1/10 or 1/15 of a dolphin. If you integrate this, they can basically, because of their physiology, they can exchange about 80 to 90% of their total lung capacity. A human can maybe do 60. So in one breath, they can exchange almost all the air in their lungs. Also, here is the oxygen content in the periphery. So this is end tidal gas concentration. And in humans, it's about 13%. Oftentimes in these animals, it's between 8 to 10%. So they seem to be hypoxic. They live with low levels of oxygen in their tissues and in their blood during most of their entire life. This is a comparison between a human and a dolphin, just to give you an idea of the differences. In a human, we breathe with an inhalation first and then an exhalation. All marine mammals, most marine mammals breathe on exhalation followed by inhalation and then a long respiratory pause. All the cetaceans do these. We're not entirely sure why they have this long respiratory pause, but one reason could be for having increased buoyancy. Interestingly, for those of you who swim or scuba dive, this is how you breathe when you're swimming. So if you go into the pool, you start swimming breathing like this. When you start swimming, you exhale, inhale, and then you continue swimming with the lungs inflated. So why is this important? So we talked about the structural properties will affect gas exchange at depth, and that will affect their ability to dive and potentially affect the risk of decompression sickness. Well, we'll come back to that. But this unusual anatomy of these animals, especially the aquatic animals like dolphins and whales that always live in water, gravity may have a huge effect on their ability to breathe in when they're on land. So we were looking at this because this could have some implications for stranding networks. This is a flow volume curve. Again, these you've seen before, they were slightly different. But here you have volume on the x-axis, you have flow rate on the y-axis. Exhalation is upwards, inhalation is downwards. This is a dolphin in water. This is when they're forcefully exhaling. So they're trained to do these forceful maneuvers. They do this because of veterinary testing, so sometimes they need blow samples from them. You see the difference when the animal is on land versus in water. So we did these measurements, and here you see the difference. In white, the animal is in water. The same animal directly afterwards beached. You see the reduced respiratory capacity, the reduced respiratory volume. So this will have significance when these animals are beached, so during mass strandings, for example. So we're trying to develop this equipment with animals in human care to then provide equipment for stranding networks so they can do direct testing of animals in the wild, so they can then assess if animals are becoming compromised. This was some data we got from a beached false killer whale. This was the animal was beached in Vancouver Island up in Canada, on the Vancouver Island. And you see in red the animal was brought into the aquarium, and this was the first day of testing. This is about 2 months later in blue, and then in the black about 6 months later. You see how the capacity has increased. The animal was also growing, but his mass specific, so the volume of air per kilogram body mass increased significantly. So after beaching, these animals may have problems opening up the lungs, and there may be some pneumonia that he was recovering from. But you can see how this tool can be used to assess respiratory health in these animals and how the respiratory capacity is increasing. So this will also help stranding networks to assess if animals have respiratory disease. We also developed this because we work a lot with animals in human care, and also, like I said, it would be good to be able, for the stranding networks, to be able to have the capacity to do lung function testing and assess health in animals. And for those of you who have gone to the pulmonologist and done spirometry or lung function testing, you've seen these before. This is the flow volume loop from a human. So this is expiration. We have this with this triangle shape, and then during inhalation we get this curve. So we have flow dependence. So we can push and push and push, but only at a certain volume can we generate the highest flow. Marine mammals are very different. You don't see that in a dolphin when they're doing maximum flow. So the tubing, the engineering inside these animals is very, very different. So they can maintain high respiratory flows over the entire lung volume. In humans, we use this, or the pulmonologists use this to assess respiratory disease. So these flow patterns change predictably with different diseases. Here you see a human with emphysema. So we've been using this in animals with respiratory disease, with certain diseases, oftentimes pneumonia. So here's a healthy animal, and here's a sick animal. So you see how obvious it is when you have an animal with certain lung diseases to be able to detect this. So now we can do detection, we can do diagnostics, and then we can track animals through time when they're treated to make sure that they're getting better. So getting back to the work by Scholander. So you remember that he proposed that this was the adaptation from all marine mammals. They had a non-collapsible upper airway, they had a collapsible chest and alveoli, and that led to lung collapse, and this was saving all the animals from decompression sickness. So we've shown that this is not true for all animals. It's huge variation, and we wanted to then try to get an understanding, having done all this work, get an understanding at what risk are these animals of decompression sickness? Are they ever at risk with repeated dives, or are they never at risk? This is some work done by Tom Berghage in 1979, and then was followed up by me in 2017, where we're trying to predict and see decompression sickness risk in different species. So what you have here is you have body mass in kilogram, and then you have something he called ED50. So this is the saturation pressure. If you're diving, how much nitrogen you have in your tissues as a partial pressure, that gives you 50% severe decompression sickness. You see that for a mouse, they need a lot higher dose. So they need a lot more nitrogen. They can dive to much greater depth before they get decompression sickness. And then this risk goes down, and this overexposure, or how much nitrogen you can have in your tissues, goes down significantly with increasing body mass. So what this was suggesting is that DCS risk is correlated with cardiac output. So this curve is an allometric curve that correlates quite well with changes in cardiac output. So cardiac output per kilogram body mass is much higher in a mouse, so they can take up and clear gases much faster than a dog, a goat, or a human. So this is all explained by physiology. So we went back to then first principles, and we've gotten all the information about marine mammals. So at what risk are marine mammals if you look at their diving behaviors? So starting off doing that, believe it or not, this is a marine mammal. Well, you have a lung, you have a cardiovascular system, you have an arterial side, you have a tissue, and then you have a venous side. So gas is taken up at the lung depending on the partial pressures. So it's all driven by partial pressures, then transported through the circulatory system here on the arterial side to the tissue. And then when the lungs are closing, when these animals are diving and the lungs are compressing, the gas inside here is increasing in partial pressure because the pressure on the outside is pressing on the lungs. So then more gas is going into the blood and to the tissue. So the animals are taking up nitrogen, which is in the lungs. So you increase the amount of nitrogen in the blood and the tissues during the dive, and then it's released when they're coming back to the surface. To make it a little bit more realistic, we added some more compartments, and we looked at different tissues. So you have fast tissues where you have a lot of blood flow and they have low solubility of nitrogen. We looked at slow tissues that have a little bit of blood flow and very high solubility of nitrogen comparatively. We added something called a shunt, so we could shunt things past the lung to look at if there are physiological or anatomical shunts. So basically, if there's mismatch, if there's no gas taken up when the blood is passing by the lung, how will that affect the modeling outputs? We took a bunch of dive records. Here you see the differences between a beaked whale, sperm whale, another species of beaked whale, pilot whale, a killer whale. And these are all, you can see here, from 0 to 1500 meters, 0 1500 meters, 0 1500 meters, 1000 meters, and the killer whales only to 150 meters. You see the dive behaviors are quite different. But what happens then if we use the model to predict nitrogen tension and potentially decompression sickness risk on this figure? So if we overlay the data that we had from the modeling experiments here, where are the cetaceans? So here are some of the shallow diving beaked whales. Here are some of the intermediate diving beaked whales, and here are the deep diving beaked whales when they're diving deep. Here's the dolphin, and here's the honorary marine mammal, which is the king penguin. And you see they're all suggesting, these data suggest that they all should have about 50% or more decompression sickness risk after each dive during their dive bouts. And apparently they don't. So what is going on? What is it we don't understand in the physiology of these animals that we need to explain to understand how they're doing this? Because if you look here, here is someone that you may have seen if you're out on the water. This is dolphins from Sarasota, and you see what they're doing here. You have time in seconds. This is a little bit less than an hour. Here you have depth in meters, so multiply this about 3, and you get feet, so this is about 9 feet. So they're not doing a whole lot. They're not diving for very long, they're not diving very deep, and when you put them on another species, the same species of dolphin in a different area, the Bermuda dolphin, you see that they're doing something completely different. So how can the same species doing such wildly different things, and what are the differences in physiology? And we did some measurements on these. So it's not metabolic rate, because the metabolic rates are the same. It is not the lung function itself, because the lungs are responding differently. The lungs are equally squishable or compliant in these different species. So we put all this data together, and also some data with work we did in turtles, and we came up with this new hypothesis where we suggest that these animals have the capacity to vary uptake of different gases, and I will try to explain this. So basically they can select by matching ventilation and perfusion. So they can select either uptake of oxygen and CO2 and then prevent uptake of nitrogen because of their differences in solubility. And I will try to explain a little bit how this hypothesis is made up. So here's some data that was published in the 1960s by Leon Fari. What you have here is, this is a little bit difficult to understand. This is ventilation and perfusion. It's basically how well the lungs or the alveoli are inflated and provide new fresh gas to the blood, or how much blood is passing by the alveoli. So you can play with these two, and you can either have a collapsed alveoli where this will be 0 and there will be no gas exchange. So you just have perfusion pass, so you just have blood passing by a collapsed alveoli and there's no gas exchange. Or you can have perfect matching where there's enough blood passing by the alveoli and the partial pressure in the blood is the same as the partial pressure in the alveoli. So you have this perfect matching. Then depending on how you are adjusting these two and how much blood is passing by or how much gas is in the alveoli, you see here that there are differences in where you can go with the matching. So if you work in a certain region, if you work in this region here, you have perfect matching for oxygen and carbon dioxide, and you have no exchange or very little exchange of nitrogen, and this would be the perfect match for a diving animal. If you can then adjust these two, you could then exchange oxygen and carbon dioxide, which you want to do because you want to take up some of the oxygen, get rid of the carbon dioxide, and have no uptake of nitrogen. So how does this work? One of the things that we detected and we suspected that these animals have, especially the cetaceans, is something called collateral ventilation. You may remember from biology, you have the airways here. Here they have the bronchi and the bronchioles, and then they end up in these alveoli. So you have these alveoli, and they're supposed to be independent. So this alveoli are not supposed to communicate with this alveoli, or this alveoli is separate from this alveoli. But there seems to be these pores, so these passages, these interalveoli passages. Here you have, these are called the pores of Kohn. These are called the pores, the channels of Lambert, and these the channels of Martin. So there seems to be ability for gas to flow to different regions through these different channels. So what we did was we had the opportunity to do some measurements on a live dolphin because he was being brought out of the water for a procedure, for a medical procedure. So this is something that is oftentimes done in the clinic in human hospitals. So what we could do is we could block one of these regions. So we blocked this region here, and then when the animal was inhaling, air could not enter this region right here, but it could enter this region, and if there's a lot of collateral ventilation, air would move over to this region through these channels. If there's no collateral ventilation, this region here would eventually collapse because there's no fresh air going in, but the air could go out. So it was just blocked in one way. So I will show you some of these data. Here you see human with collateral ventilation, human without collateral ventilation, and this is a dolphin. So on the bottom here in blue, you see each blue peak is a breath. And a human with collateral ventilation, this is flow out of this blocked region. So this suggests that there is another pathway, there's another route of gas into this alveoli. So one of these channels. Here's a human without collateral ventilation. So you see how the alveoli is slowly collapsing with each breath. So it's getting smaller and smaller because you can't get new and fresh air through these channels. Look at the dolphin. This dolphin has immense amounts, so we couldn't measure how much collateral ventilation this dolphin had. So it seems like rather than having alveola like we think of these grape-like structures, it's more like a big mesh, like an open bag. So it seems like each half of the dolphin lung or the cetacean lung is open, so they can flow gas through each of these right and left lobes. So I'll come back to that in a little bit when we're putting all this together. Two other clues were hypoxic pulmonary vasodilatation. This is something that is specific for marine mammals. In humans and terrestrial animals, you have hypoxic pulmonary vasoconstriction. Think about this. If you have a collapsed and hypoxic region, you don't want to waste any blood to go to that region. You want the blood to go to the open regions because you want to exchange the gas. Marine mammals seems to have the complete opposite. So when the lungs are hypoxic, you dilate the blood vessels. Basically, there is more blood going to that collapsed region or to that hypoxic region. Also, there seems to be condition control of heart rate, and I will show you some of the work we've done in dolphins. So this has been shown in harbor porpoise and also in sea lions, that these animals, when trained, they seem to be able to have this amazing capacity to vary their heart rate. This is how we do this on dolphins at a facility. So this animal is trained to turn upside down voluntarily. We put on the suction cups that then we can measure the electrocardiogram. And then we train the dolphin to hold its breath for varying amounts of time while we're measuring the heart rate. If you've taken physiology or remember something called the dive response, basically what happens in all animals when they hold their breath, and especially when you hold your breath in humans, if you dunk your head in water, you have this dive response. The heart rate is changing. So here you see on the x-axis, you have time in seconds. You have instantaneous heart rate. So this is the heart rate between each beat. Here's the animals breathing at the surface. Here we ask the animal, turn around, hold your breath. So the animal is upside down. As soon as it takes his last breath, we start the beginning of the breath. And you see how the heart rate is going down. So this is supposed to be a reflex. This has been reported since Scholander that this is supposed to be a reflex. This is supposed to happen every time that you hold your breath, and it's supposed to be the same every time. So you're not supposed to be able to vary this. So what we thought was, let's train these animals. Let's show them a sign for a short dive or a different sign for a long dive. Well, let's have a completely different signal for you do whatever you want. You can hold your breath, you cannot hold your breath, and we're just going to measure your heart rate and see what happens. So when we show them the sign, we get the heart rate that I showed you before. When they're not shown a sign, this is what happens. There's hardly any change in heart rate, and then eventually it starts to decrease slightly. And it's extremely variable depending on the dive duration, depending on the dolphin, depending on the day. This varies all over the place. While when they're trained to look at the sign, most often we get a rapid decrease. It seems that depending on the condition, depending on what the animal is anticipating, they adjust the heart rate and the blood flow. So let's see if we can put this together. Remember the CT images. So this is the lung. This is one lobe of a lung. This is a terrestrial animal. And when they're compressing, you get a graduation. The entire lung is collapsed gradually. In a marine mammal, you remember that gravity dependence. So you get an open region, you get a collapsed region. Because the hypoxic pulmonary vasodilatation, most of the blood will come to this region. And because of the conditioned heart rate response and their blood flow, they can adjust how much blood is getting to the lungs when they want it. So they can adjust this ventilation and perfusion to be right in that sweet spot where they can exchange oxygen and carbon dioxide and no nitrogen, and they're therefore avoiding decompression sickness. What we believe is when these animals get stressed, when they're exposed to something that is new and novel, for example, Navy sonar or underwater blast from oil exploration, this mechanism fails. When turtles get caught in fish nets, a similar mechanism fails in them. And then a lot of blood comes to the upper region and they take up a lot of nitrogen and they get decompression sickness or these gas bubbles. Okay. That was my long spiel. Hopefully I haven't been too long, but I'm extremely fortunate. I work with some tremendously gifted people and collaborators, and I worked in a lot of places, and well, I wish I could come and see you guys. We do a lot of work in Sarasota. If you're interested in participating, volunteering, and working with dolphins, then I would contact the Sarasota Dolphin Research Program, and they do sometimes take on interns, and they've been there for 50 years and do a lot of good in the Gulf of trying to understand the impacts of human impact on these species. So thank you very much. If you're interested in some of this research, you can follow us on these websites. So I will leave this up a little bit. I don't know if there are any questions or...
Can I just hop on and say thank you? That was a really great talk. I can either manage questions or Brad can, but normally in seminars when we're in person, we really like to have the students answer questions or ask questions first. So I'd like to open everything up for student questions first, and I have one announcement before we move on. So today MSAC is doing a trivia. evening at, it starts at 4:30, so in 15 minutes. And even if you haven't signed up for a team, you can join in. And I'm going to put the link to that Zoom in the chat right now. So I'm going to get off. Brad, do you want to manage questions?
I'm happy to do that.
Excellent. Thank you.
Actually, I've never done that before, now that I think about it. Let's see. I don't see the hands going up. Let's see.
So I click
All right.
on participants icon. That should list all the hands that are up, and the ones on top, first hands that went up.
Okay. I think I see a question from Brianna. You want to go ahead?
Yeah, I unfortunately had another meeting, so I was only able to come in at the end there. It seemed like a really cool talk, and so I apologize if you covered this. But I was wondering, I know when it comes to a lot of hypoxic, non-air breathing marine animals, they do a lot with kind of their blood chemistry to adjust their kind of ability to deal with low oxygen. And I was wondering, it's not my area of expertise. I have a memory that diving air breathing animals do that as well, but I was wondering how that affects their kind of ability and prevalence of getting the bends, basically decompression.
Yeah. That's a great question. So there's some evidence that increased carbon dioxide will increase risk because it diffuses much faster, and that will basically seed. So it will go into the bubble and then initially cause growth. That has been shown in, I think, in the 50s in some animal studies, and there's some evidence in some of the stranded animals that may also be a potential risk factor. Some of the work that we've done with someone put up, Randy Wells here, yeah, he's the director of the Sarasota Dolphin Research Program, and I've actually completely forgot to acknowledge them because they've been integral in some of these studies. We did some work, and there's also been some work in other species. We showed the difference in hemoglobin and hematocrit in deep divers versus the shallow divers, and basically they have a lot more red blood cells. They can pack a lot more oxygen into the blood. So that's one way of doing it. In most species of mammals, at least, not in the turtles, they do have buffering capacities that are better, but they're not suggestive of them oftentimes doing anaerobic metabolism. So they don't seem to, they have the capacity of doing it, but they're not pushing themselves, because the assumption on the hypothesis is that will cause acid-base balance disturbances, increase lactate, and that eventually needs to be basically reversed back to pyruvate and then go back into the regular pathways. So there is some disagreements with that, but the general conclusion is that most of them are not anaerobic using a lot of anaerobic metabolism.
Okay, thank you.
Yeah.
All right. Come on, students. Anybody out there? You guys must have some questions. Let's see. We got Alec Timpe. You want to go first?
Sure. I have a quick question. So I know that decompression sickness is an issue in humans when you dive for prolonged periods, but is not usually, or I assume it's not as considered if you're doing free diving. You don't hear about it as much in free diving.
Yeah, that's changing.
It's changing? Okay.
Yeah. Yeah, there's been quite a lot of reports, and the more people are getting into free diving, it's becoming more and more of an issue. There seems to be more cases of it. I think there are other things that are more problematic, like the pressure on the lungs and lung damage itself, but there's a brilliant study by a Danish physiologist from the 60s, I think, where he basically caused himself to get decompression sickness by diving repeatedly, and that was the whole intent of the study. So he dove repeatedly, I think, for about 20 times in a tower, in a diving tower, just to see if he could, with repeated dive, accumulate enough nitrogen. So it's a question how these. So this has been the whole thing. There's not enough nitrogen in the lungs for them to ever get enough nitrogen into the tissues. But if you look at, it's sort of like a savings account, and I don't have a lot in my savings account, but continuously saving up, well, you need to save more than inflation. Eventually you will actually have quite a lot of money. And it's the same thing with the way that these guys are continuously diving, and in these bouts when they're diving, there seems to be, with our best knowledge of physiology, enough accumulation that especially in these deep diving and these large species, that there seems to be some concerns.
All right. Thank you very much.
All right, Lisa, do you have a question?
Yeah. I just completed my decompression diving, so this is super interesting to me, like on many levels. But this might be a really juvenile question to ask, but like, do the other gases affect them like we would get narked, like at certain levels? Does that happen to animals?
I think so. When you talk to these free divers that do these, they dive to 100 meters on one breath, they talk of euphoria when they're diving. So what happens is the lungs are compressing, you get additional oxygen, so you get high levels of oxygen, so you may potentially have risk of oxygen toxicity. You get high levels of nitrogen being pushed in. So there is this, how do they avoid nitrogen narcosis? How do they avoid high pressure nervous syndrome? How do they avoid some of these things that we don't know a lot about? And there's basically is Jay Dean, and he may be the only one nowadays up at the Tampa campus that are studying these things. It's what are the effects on these high pressure gases on these animals? There's basically nothing. There is some studies. There's been, I think there was one study by an Israeli group earlier this year, but we know nothing. We know nothing how they do this.
Well, that's super interesting. Thank you for the talk.
All right. Any more students? All right, we'll open it up to anybody that has a question. All right, Maya.
That was a fascinating talk. Thanks. I am a microbiologist, and so I know nothing about like animals or lungs, but I was wondering how does their like flexibility of these organs change with age? And do you expect to see, are there like predictable profiles like with their breathing patterns as they age?
Oh, this is a great question. We don't know. And this is something we're trying to do now. No one has ever been able to do lung function testing on these species before. So this is one of the things we're trying to look at. How does it change with age? How does it change with this disease? What are some of the predictive patterns? And especially with the concerns of animals in human care are there things, if these animals are adapted to do certain things, and I think there's been some evidence in birds that some birds need to fly. This is a physiological thing they need to do. Maybe these animals need to dive. This is a physiological thing they have to do. Then that continuous compression, collapse, and reopening of the lungs, maybe that's an important part. Maybe we need to do something with these animals that are under human care to keep this going. So we don't know, but this is a great question. A lot of these things is driven by welfare of these animals that are in human care. But
interesting. Thanks.
Excellent question.
All right, we have a question from Naha Murphy.
Yeah, since the solubility of gases changes with temperature, is there a temperature dependence that goes into this lung function of the mammals? I don't know, it just might be a silly question.
No, it's not. No, there's been some beautiful work with people in Wilmington, and they looked at different solubilities in different types of fats. So generally the nitrogen is much more soluble in fatty tissues versus blood and muscle, for example. Some of these deep divers have waxy esters, and they seem to have very different solubilities. We don't know how temperature is affecting this, and this is something we're trying to look in with NOAA at the moment with the turtles. So the turtle story is a global epidemic that they get caught in the nets, they get pulled quickly to the surface, and then they get decompression sickness. Oftentimes the turtles are alive at the surface, they're tossed back into the water, and potentially as much as 30% of those turtles die due to complications. So these guys are not accounted for in the fisheries mortality. So you have an underestimation of the mortality rate. So we're trying to understand this better in the turtles. And the turtles, they follow because they're not homeotherms, so they're not having the same temperature always. They vary it with temperature. So it may depend on the season, it may depend on the depth they're going to, and this is a really important question for that purpose. So that may affect the risk in different directions depending on what they're doing. But we don't know.
Yeah, thank you. That's really interesting.
Thank you. Catherine?
Oh, hi. Great talk. So I'm a geologist. I'm not a biologist. I'm going to ask a really basic question. I'm wondering, because you compared marine mammals to land mammals so much, I was wondering what the evolutionary benefit of deep diving is, trying to think back as to how they developed or why they developed these diving habits and these lungs that accommodate it. And also going back to what you said just now to Maya about how it was like physiologically compulsive, I'm wondering how much of it is learned from like their communities.
Yeah, those are great questions. So the first one is, I would say food, because that's a simple answer. Food avoiding predators. If you can dive to 3,000 meters or 2 miles down, which is the deepest dive, we've measured a dive. I think the deepest is 2 miles, and we just measured a dive for a beaked whale 3 hours and 40 minutes. So holding your breath that long and diving that deep requires certain physiology, and what they do is they hunt for certain prey that are at those depths.
It seems like a lot of stress for food.
Yeah, for them, yeah, but that's because I think we are so anthropocentric, because we think about this is really stressful for me. For them, maybe being at the surface is stressful, because oftentimes these guys they're divers, they're surfacers. They just come to the surface for one purpose, that's exchange gas, and then they dive again. So a lot of these species, I think the beaked whales may be predator avoidance and then finding food. So they're trying to, there's some beautiful work done by a group in Denmark, a group in Scotland, and they're doing a lot of work in the Canary Islands, looking at how they are diving together and looking at how they're trying to outmaneuver predators at the surface, basically killer whales. So they're doing certain things and they're coming up at certain angles, because they basically, if a killer whale sees you going down here, if I do this versus coming straight back up, the killer whale is going to have to search a much greater area to basically find me again.
Cool. Yeah, and I asked about physiologically compulsive or learned.
I don't know.
Okay.
This is a great question too, and the more we can, with biologging tools and with physiologging tools, we can start to answer some of these questions. Biologging has been around for 40 years maybe. The ability to archival tags recording some physiological meaningful data has been really slow, but there's some beautiful work by a guy in Scotland. He's been measuring using near-infrared spectroscopy on seals, and he's also got something done on humans. So being able to understand what's going on inside with not just heart rate, but also how oxygen is partitioned in different tissues at different times of the dive, it's really going to help us understand how things are going on and how they are learning this. And this is something that we could learn how to do with animals in human care, because we can validate the tools, we can do controlled experiments, and then bring this out and study wild animals.
Cool, thank you.
All right, thanks. Christina.
Hi. So you had shown that one slide where it was looking at if the gas was coming in straight through the alveoli or if it was going in through those side channels.
Yeah.
And I was wondering why some people would have that and some wouldn't, and if you see those interspecies differences in other animals too.
Oh, that's a great question. Do you want to do physiology?
Yeah, I'm actually in Brad's lab.
Don't steal my students.
It's one of the things we're trying to answer. In humans, this is a pathology. So they do these measurements before they're going to do lung surgery. So sometimes people have a portion of the lung where they need to take the lung out. So they do the measurements to try to understand which portion of the lung do we need to cut out, because you don't want to cut too much, because you want to keep as much lung as possible. In the marine mammals, this is mainly in the cetaceans. When we do the initial tests we did, because someone suggested they have collateral ventilation, this is how they avoid decompression sickness. So then we started to look at this in cadavers, so animals that had drowned asphyxiated in fish nets. Then we had an opportunity to do it in a live animal. So we do think the current data suggests that there's minimal collateral ventilation in pinnipeds, so in seals and sea lions, which makes sense. If you remember the lungs on the cetaceans, you have two lobes. You have one half on the right side, one half on the left side. So this lung is a complete unit. That lung is a complete unit. In all the other species, you have these lobulated lungs. So you have a separate piece here, and then you have another piece here, and then you have another piece here. So no matter what you do, these are sort of going to be independent of each other. So you can't really have that connectivity in each of the lobes. This is about as far as we know it. Not most, but some species, land species, do have this, but you really need to increase the pressure inside the lung before these pores open up. So it's not generally open. In the dolphins, the data I showed was there was so much collateral ventilation that it's sort of, you can think of it as the terrestrial mammal having basically a cluster of grapes, and each grape is like an alveoli. In the dolphin, it's just like an open bag. It's sort of big bag hanging, and then you can just exchange the gas inside. So it's basically one unit.
All right.
Thank you.
Yeah. David Nahr, you have a question?
Yes. I wanted to ask you, this is kind of a far reach, but you keep talking about lungs, and I keep thinking about COVID-19.
Yep.
Is there any possibility that your research, maybe you already mentioned this and I missed it, that your research may have relevance on COVID-19 research?
Yeah, something completely different that we've been working with. I work with an ecologist at Duke, and the current research seems like the way that these guys are avoiding hypoxia and inflammation are related biochemically and immunologically, but it's completely different in these species versus in humans. So in humans, every time you're hypoxic, you get inflamed. But imagine these animals getting an inflammation every time they dive. That would be completely sort of counterintuitive. So it seems like in at least in the dolphins that we measured, these two are separate and they function very differently. And this seems to be one of these cytokine storms that they've been talking about. Seems to be related to that. And there's some other evidence, some people that have looked at immune function in these animals that seems to be quite different. So this is one of the things I've been sort of stamping my foot about, because I think these animals are uniquely, both marine mammals, turtles, a lot of these marine species live, and I think that's a great question before is, but it's so stressful. Well, for us it is, for them it's not. But they're doing things that has clinical implications for us. Acid-base balance disturbances, hypoxia, collapsible lungs. I mean, collapsible lungs is a huge issue for prematurely born babies. So if we can understand how these lungs are collapsing and opening up, not only can we help prematurely born babies, we can help people during surgery when the lungs collapse and you put positive pressures on the lungs with potentially the risk of the lungs being damaged. You hear a lot about people being intubated during COVID-19, and that has an effect. So if we can understand how the lungs could stay open more easily, that a lot of these things could have clinical implications. I haven't convinced NIH yet, but maybe one day.
Well, we'll keep working at it. I'm sure you will.
Yeah, we'll see.
All right. We've got time for one more before we let everybody go to the trivia night. Anybody? All right, I'll ask you a quick one. What are your thoughts on the mammalian dive reflex in humans and other terrestrial animals? I learned it as a sort of evolutionary vestige or something, and still doesn't seem to have much practical application for us.
Yeah, no, I'm sort of, I think it's more like let's try to avoid hypoxia. I think it's not just related to diving. There's been some Peter Huchachka, that was Ken Story's supervisor at UBC in British Columbia, made some suggestions to that this is not just related to diving. It's more of a generalized hypoxia response that you see it in fish that are being brought out of water. You see it in all species that have been tested, and it's not just for diving. I think that there are adaptations that some of these species have that are dive adapted is manipulating this dive reflex. So I don't think that it's a reflex. I think in these species, they have much more capacity and they have much more leeway to being able to move that around.
Yeah. Very good. Well, thank you so much. That was a fantastic talk. I really appreciate it. I don't know how to get everybody else to let you show their appreciation, but
thank you, guys. I got to rush to the airport, right?
Right.
Well, we can all open our mics and have a grand unison applause. So thank you.
Thank you, guys.
Thank you very much. We really appreciate it. And thanks for being our first virtual guinea pig.
Well, I'm glad to be it.
All right. Well, take care. I'll be in touch.
Yeah. Thanks, guys.
Enjoy dinner.
Oh, I will. Bye.